Nearly every organism that is made up of many cells begins life as the single cell of a fertilized egg. That single cell divides over and over again until eventually an embryo is formed that is made up of trillions of cells of many different types. Over time, the embryo develops into a baby. And even at a very young age, some of the baby's cells begin to wear out. In fact, in a typical human being, every second of every day witnesses the death of about 50 million cells. Therefore, new cells must be constantly produced to replace old, dead, and damaged cells. For cells to reproduce themselves, whether in a developing embryo or in a fully grown adult organism, certain definite steps must be followed to assure that the new cells will contain exactly the same genetic material or genes that were originally present in the parent cell. And the essential process underlying the reproduction of cells is called mitosis. Mitosis is defined as the duplication and division of the nucleus of a cell and its chromosomes during cell reproduction. Scientists recognize 4 distinct stages of mitosis. First is prophase, second is metaphase, third is anaphase, and fourth is telophase. An average of about 6% of a cell's total lifespan is spent in these 4 stages of mitosis, while the other 94% of its life is spent in a stage that is not considered to be a part of mitosis, called interphase. Because the events that occur in interphase allow mitosis to take place, let us begin our examination of mitosis by taking a closer look at interphase cells. Interphase is defined as the period of a cell's life cycle between one mitosis and the next mitosis, the period of cell growth. As can be seen in this microscopic image of an onion root, some cells are dividing, but the nuclei of most of the cells are round and intact. These are interphase cells. Average human cells, such as these that form connective tissue, spend about 19 hours in interphase and only between 50 to 90 minutes in mitosis and cell division. However, there is considerable variation among different types of cells as to how much of their life cycles are spent in interphase and in mitosis. For example, this human nerve cell rarely reproduces. Instead, it can remain in interphase for up to 60 years. Because nerve cells rarely reproduce, even to replace dead or damaged cells. Nerve damage, such as results from strokes or spinal cord injuries, is usually very serious. On the opposite end of the scale from nerve cells are cancer cells. These purple-stained cancer cells, which stand out clearly against a background of red blood cells, were taken from a leukemia victim. Cancer cells like these do not remain in the interphase state very long. Instead, mitosis and cell division continues at a furious pace. In fact, cancer can be thought of as cells that never rest, that divide over and over again, never remaining very long in interphase. Even though it is often thought of as a resting state between cell divisions, a lot is actually happening inside the nucleus throughout much of this stage of a cell's life cycle. During interphase, deoxyribonucleic acid, or DNA, the enormous molecule that forms the cell's genes and that holds all of the cell's operating instructions, duplicates itself. Deep inside the nucleus of interphase cells, the 2 DNA strands unwind as new copies of the DNA are created. Scientists refer to this process of DNA duplication as replication, because exact replicas of the DNA are produced. The replication of a cell's DNA makes it possible for 2 complete sets of biological instructions to be sent into the new daughter cells. During mitosis, DNA is found within chromosomes, but chromosomes don't actually exist during interphase. Instead, DNA and protein form the grainy threads we see here called chromatin. The presence of chromatin is one indication a cell is in interphase, for only with the onset of mitosis does the chromatin mold itself into the distinct shapes of the chromosomes seen here. Scientists still do not understand the exact role that chromosomes play within the cell, but it seems likely that chromosomes are the best way to package DNA, and consequently genes, for shipment into the newly forming cells. The chromosomes seen here, taken from the salivary gland of a fruit fly, provide a convenient material for scientific study because they are 10 times longer and 100 times thicker than normal chromosomes. Working with these cells, scientists have discovered that the dark and light bands we see here are the actual location of genes on the chromosomes. And their studies have also shown that when a particular gene on one of these chromosomes becomes active, the spot on the chromosome where that gene is located takes on a strange, puffed-out appearance. During mitosis, the appearance, movement, separation, and disappearance of chromosomes is of critical importance to the cell reproductive process. In fact, by recognizing what is happening to the chromosomes, we can identify each of the 4 stages of mitosis.
During the first stage of mitosis, known as prophase, threads of chromatin found in the nucleus of interphase cells condense into the compact structures seen here, called chromosomes. The chromosomes contain most of the cell's DNA and are where the genes are located. As prophase continues, the nuclear membrane disappears and tiny tubes called microtubules gather to create a structure called the spindle. In animal cells, the spindle takes shape around 2 structures called centrioles, not found in plant cells. It is important to realize that during prophase, the chromosomes are actually doubled and are made up of 2 identical strands called sister chromatids. The sister chromatids exist because the cell's DNA was replicated during interphase. A special structure called a centromere joins the 2 chromatids together. During metaphase, the second stage of mitosis, spindle microtubules attach to the centromeres of the doubled chromosomes. Metaphase ends when all the doubled chromosomes have arranged themselves on the equator of the spindle. Anaphase, the third stage of mitosis, begins when the centromeres split apart. Then, with the help of the spindle microtubules, the sister chromatids pull away from each other and become separate chromosomes which swiftly move in opposite directions. During telophase, the fourth and final stage of mitosis, the new nuclei make their appearance as nuclear membranes form around the clumps of chromosomes. As the new nuclei develop and the chromosomes change back into chromatin, cytokinesis, or cytoplasmic division, occurs when the cell membrane pinches off. At this point, cell reproduction has been accomplished, and there are now 2 new daughter cells, each of whom possesses the same genetic information as the parent cell.
The stage of mitosis seen here is called chromatin shortens and thickens to form these distinct structures called DNA is replicated during the stage between cell divisions called true or false. The division of the cytoplasm following mitosis is called cytokinesis. The first stage of mitosis, the stage when the nuclear membrane begins to be reabsorbed into the cell, is called